Pretty thighs, shawty, she a dime. Demon girl, evil eyes, she be telling lies. Slim waist.
I just mentioned that recently you managed to cultivate what is described as mini brains that have their own optic hubs. So this obviously is a great deal of technical and biological know-how. So what was your intended process for this undertaking, and what was the process behind this feat?
That's a great question. So as I mentioned to you, we were simply generating brain organoids. Let me explain you what are brain organoids. So there is a history one should understand. So people were studying mostly brain development in the 2D cultures. And then there were two important people we should credit them. We should offer credit. Yoshiko Sasai and Flora Vaccarino. Yoshiko Sasai is a Japanese professor. He was the first time to show that he can grow those brain-like tissues along with the Flora Vaccarino from Yale. Unfortunately, Sasai is not with us in these days. He passed away, unfortunately. So, and these were the two pioneers to bring this technology forward. And then many labs from Stanford, Sergio Pasca, Lancaster, Madeline. So they all moved this field further. Great work they have done. So we were studying only on our focus. Our contribution was only on making the brain organoids. Now, the brain organoids means that it's not only a simply collection of some neural cell types, but the stem cells with ordered organization of brain-like phenotype, brain-like characteristics, which is the amazing property of the stem cells to organize such a in vivo-like structure. And we were only studying about the microcephaly, what causes the small brain. Then I was looking at one day, it was around 2017. So I was looking at literatures, in 1850, people have done amazing work because those are all mostly embryologists. So they were looking at different animal development and including human embryo development. And they looked at, already 8th to 9th week of human gestation, you start to see those optic vesicles coming from the brain. So, and if you go to Carnegie Mellon embryology, resource, it is well described in human brain development, the primitive optic vesicles, they start appear around 8th to 10th week of human embryo development. So when I was sitting down in my office, I was asking myself, wait a minute, if these brain organoids are really useful tissues, they should be also able to assemble these optic-like structures, optic vesicles, but not many, not one, but precisely two. And if they don't form, they are basically stupid tissues. They don't help us. It's just a blob of cell types. And then I looked at the literatures, then we found critical factors that you are supposed to, that they are important for optic vesicle, formations. And then we found some of the factors. One of them is retinoic acid, which you always identified. It's a vitamin A derivative, which is important for the brain development in the early stage. And then we used this compound in different time points, and the one time point it started generating, these two nice dots, and that was a starting time point. Okay, good, something is very interesting. Now we need to develop our experiment around this area and try to find out. And then this took almost 4 years to work on this problem. And then we started developing it further. Then we identified eye kind of cells, eye-like cells, like the cells that you see in the human eyes. We found them. We found these axonal projections, and they were light sensitive. It was totally amazing experience.